What treatment or therapy are you most proud of in your career that you help advance?
My father, before he passed, was on. It can do it faster than a human can do, and it can catch things that a human wouldn't catch. You know, there's 70,000 new viruses found.
Going to meetings like AAPS, the reason that you get together, what you really care about is getting drugs to market to help people and making them effective and making them safe. If you could go back in time and visit your 25-year-old self, what would you say to yourself?
I would probably say...
Welcome to the BioTalk Unzipped podcast, where we unzip the stories behind medical progress by sharing the latest and greatest advances in biopharmaceuticals and medical technologies in a fun, entertaining, and enlightening format. And now your co-hosts, Gregory Austin and Dr. Chad Briscoe. All right, so we are back at AAPS at PharmSci 360. And Mark, I'm curious, what treatment or therapy are you most proud of in your career that you help advance, and what was your role in getting it over the finish line?
That's a tough one, mainly because, at 30 years of BMS, when I left, every single drug in their pipeline, because I had been running the bioanalytical department, I had some hand in.
That's cool, though. Wow. I mean,
yeah, it is. That's amazing.
Amazing. Yeah. Wow.
And I look at it right now. I'm still on 2 of the drugs I helped develop.
Oh, wow.
Wow. Yeah.
There's a personal connection right there. Yeah, it's literally helping you. That is, yeah.
My father, before he passed, was on 3 of the drugs I helped develop. So, it's that type of thing that, and you talk to other people and things, but when you look at it, the diabetes drugs are important. But then you look at all the oncology drugs, that, the Orenches and those types of drugs that are really the lifesavers.
Right.
So,
and I've heard many times from people like you, Mark, and Chad, going to meetings like AAPS, the reason that you get together, besides hearing about new technologies and new sciences and better ways of doing things, is even though you're working in competing companies, maybe with even a competing therapeutic, that what you really care about is getting drugs to market to help people and making them effective and making them safe, because you know what? It's going to be our kids, it's going to be our parents taking those drugs. And here's a perfect case study. You and your father have taken drugs that you have had a hand in developing. So I think that's really, really cool.
We come here and we share our science, right? We talk about on the bioanalytical side.
Absolutely.
What our methods are. When it was, both of us were there with the early days of LC mass spec. How do we measure things better? What were the problems? And everybody learned from each other, and our methods got better. We got over all of the interferences. We got past what was Muhammad Jamal's term for the super fast chromatography.
Right,
And we missed out that, yes, all the interferences were there and were saturating out our signals, right?
Yep.
And, we learned, yes, we needed chromatography, and we got through that. And then we're now in the age of high-res mass spec.
Exactly.
And we're into proteins and all of these things. And we just continue to advance the science by sharing our insights. And, if we weren't doing that, we'd still be back in the days of HPLC UV things, and dealing with, assays that, we got an answer, but it's not the best, and it's slower, and we're not advancing the drugs as quickly as we could be. And, it's the same on the immunoassay side. Immunoassays of 20, 30 years ago, didn't have the quality reagents, didn't have the technologies that are available today. We didn't understand how to make the assays better. We didn't understand immunogenicity the way we do now. And it all comes from people getting together.
Right.
How many white papers have you been on, have I been on?
Oh boy,
that all
have we been
come
on
from-
together?
Yeah,
Yeah, for
they
sure.
come
Yeah.
from the
Yeah.
collaborations and
Yeah.
discussions of this is the best science and how we
Absolutely.
share it.
Yeah, absolutely.
And that's just the bioanalytical side. I mean, APS has, you know, the preclinical side, the pharmacologists, the clinical PK people, the PK/PD people, the formulation people, the CMC analytical, the manufacturing people. You know, all of these sciences come here and share their science and help advance each other.
And that's one of the things that I've really tried to take advantage of over the last couple of years is not just the bioanalytical interactions, because I can reach out to those folks anytime, really, and do. But it's the opportunity to meet with people that are on the manufacturing side, you know, that are CDMOs versus bioanalytical CROs, and learn and interact with them. I've seen a few former colleagues from other companies that are now in that other side that I would only see at this meeting, and then the opportunity to talk to them about what do they got going, how's that piece of the industry changing. That's an interaction I don't get anywhere else, really, and I do value that. Yeah.
And having worked in pharma for so long, you know, when you were on drug development teams, you got to see the whole panorama of all the pieces that go from discovery into the final product. When I moved over to the LabCorp CRO world, you know, you see silos of slices, and you know, people don't really understand how all the pieces fit together. And you know, I really hope that people who come to AAPS and other conferences, well, there aren't too many like AAPS for the breadth of science, take advantage, as you're talking about, of seeing some of the other sciences, so they see how it all fits together, and how some of the interactions are. Because we generate PK data or data for biomarkers that go into PD models. But do you ever go and listen to one of their talks to see how the data is interpreted?
No.
And
to be perfectly honest with you, and I should, right? It's but I'll just say it, because you asked.
And I mean, I'm a pharmacologist by training, so I had to go through all the PK courses and understand all that. So I appreciate it. You know, I learned mass spec on the job. So, you know, you could tell me all about the source and all of the ion transitions and everything that happens, and okay, I'm going to listen very intently.
Right.
But you sit me down in a meeting and then somebody starts talking about the biology, and I get that, and I get how everything goes into the biology and the PK much better. So, you know, I think it's just an overall having that integration picture really helps the scientists understand where they fit and how they contribute to getting the drugs.
And I think I think that's an interesting message, thinking about education and when students come along, because I was my education was very single track analytical chemist, right? I mean, I'm moving along in my PhD, and it's chromatographic theory, and it's how do the quadrupoles work in a mass spec, and the math behind it, and all those details that, to be perfectly honest with you, once I get into industry, I'm going, man, I wish I had more biology and organic chemistry expertise to really understand what's going on. The instrument side of it, unless you're making the instruments, the instrument side is, how do you push buttons and run software? And it's even more so now. I don't think we need this. In some cases, yes, for sure, but in a lot of cases, we need to hire people that understand the biology and the chemistry more so than the details of the instruments. So, interesting thought on that. Yeah.
Nice. So Mark, if you could go back in time, imagine yourself as a 25-year-old and visit your 25-year-old self out of graduate school, what would you say to yourself and why?
I would probably say take more risks. I have always been somebody who didn't want to take risks in my career, but looking at some of the opportunities of not taking a career risk. I mean, 30 years of BMS, in all the bioanalytical track other than one time where I was given the opportunity to work in a CMC lab for 8 months, just because it was a career development for a future executive track.
Right.
I could have gone and branched out a little bit, maybe into the discovery space or into the talk space based on my background, or into the PK realm, and just gotten a little bit broader experience there, and see if that would have opened up other opportunities. Would it necessarily have helped out? I don't know, but in the end, I could have taken a little more risk.
And I think that's good advice. Yeah.
Right. No, I mean, I think we saw a very similar response. I'll miss some. Maybe you can think of some, but Russ Weiner embraced the fear. Russ Grant was talking about taking the risks, and Russ, it seems like he is a risk taker, but he had a similar thing. Andy Vick, another guy that was talking about the same sort of idea when we posed a similar question to these guys. So over and over again, we're hearing the same theme, and I hope our younger listeners are hearing it and thinking, what's that mean, and how could that mean?
There is a different approach in the younger generation, right?
Yeah.
They are there for a company for a while, and if they feel the fit, right, they're staying in a position.
Yeah.
But they are looking for things that give them whether it's an emotional fit that is better for them, or an advancement, or small biotechs, that innovative entrepreneurial thing. I think a lot of the young scientists are seeking out different opportunities that way, and moving every year to 2 years, which back in the day, close to 40 years ago when I started, if you moved companies, there was something wrong with you, right?
Right.
Now,
Somebody doesn't want this guy.
Yeah.
And your father's generation was even more so. You stayed at the place that you had your first job, and you retired 50 years later from that same company. Yeah.
And now it's not viewed that way.
So would you advise young PhDs coming into the marketplace for the very first time? Do you think it's better for them to go to a smaller company or a larger company to start their careers?
The advantage of smaller companies is that you're asked to do many different things in not a very limited area, because they don't have a lot of expertise, right?
Right.
And so you may be in a lab and you may be doing in vitro tests for enzymatic results, or cell tests, and then you may be doing bioanalytical testing out of animals and things like that, and you're getting a broad variety of things, and you may be asked to then do the PK analysis coming out of it. Whereas if you go into industry and you take a bioanalytical position, okay, this is what you're going to be doing. It's a much narrower type of thing. So you have to be thinking about, this is where I'm coming in, and where do I want to go?
I think it's pros and cons, and I think it's also based on the personality type. I think some people are much more open to, trial by fire. They're going to, go figure it out, and they're resourceful, and they kind of thrive off that energy. And there's some people who just would panic and be like, if I don't have someone to kind of mentor me and walk me through step by step, I feel like I'm going to fail. So I think there's that part of it as well. And I think I've seen people navigate through their careers where they might start on one side, and then they kind of go into, all right, now it's time for me to jump in and do something more risky or outside my comfort zone, and I'm going to jump into a startup.
Yeah, I think some of that also comes from who your advisor was, say in graduate school.
Right.
My advisor was really hands-off, and so I kind of had to find my own way. And when it came time to get out of graduate school, I already had a family, and it was like I couldn't afford to live on a postdoc.
Yeah, right.
So it was like, all right, I got to get into industry. And so that led me down that pathway and type of thing. And some of my conservatism was because, yes, I've got a family to support, and not taking some of those risks. But I definitely would have, even inside of BMS, said, look for other opportunities.
And I've said the same thing several times as well. I had a family when I was quite young, and so a lot of the decisions are driven by having a safe, secure space for that family. I like to think of it as a noble decision to go forward. I wouldn't change a thing. And I know you're a devoted family man, and that's wonderful. Yeah. So it's really important.
Excellent. Well, Chad, it's time for new news.
New news.
Yes.
All right.
Published also on October 16th by Jonathan Gardner. This is about Novacure. They won FDA approval for electrical field device in lung cancer. So this is their product is called Optune Lua. It's a wearable device that creates electric fields to disrupt cancer cell division for non-small lung cancer. In a phase 3 trial, patients using the device alongside chemo or immunotherapy had a 26% reduced risk of death. However, there is a little bit of controversy on the way the trial was run, because the first, typically the first line treatment is going to be the immunotherapy. And that's where the controversy kind of stems in, because you've got the immunotherapy, you've got chemotherapy, and I don't know that it was done exactly the same way every time. So this phase 3 trial included both the immunotherapy, the PT1 drugs, and chemotherapy. And even though the PD1 drugs like your Keytruda are typically used as the first line treatment, the chemotherapy is the standard for second line after the cancer has progressed. So the critics argue that the trial should have only been tested with chemo to align the typical real-world second-line treatment practices. And so additionally, the trial subset where Optune Lua was combined with chemo, the increase in survival, which is 2.5 months, compared to the chemo alone, was not statistically significant, kind of raising some questions about, is this really beneficial in that scenario? So despite the mixed views, the approval is expected to boost Novacure's revenue, and additional trials will help provide some further clarity on its effectiveness. And this FDA approval is not the first one for Novacure. They've had one in 2011 for glioblastoma that showed some nice effectiveness, and another one since then. Now, I know you haven't read into this, Mark, and maybe you have, but do you think this sounds like the FDA jumped the gun a bit, based on the way the trial was run? The side effects are minimal, but there's an argument that it's only incremental improvements, which isn't necessarily a bad thing, right? But are we giving patients false hope based on these results?
That's a lot of different questions put in there.
Yeah.
Sorry about that.
Yeah.
No, I mean, the question is, is the survival real? And is the study design, that to me is the real issue, right?
Right.
It's not comparable to anything historical. So how do you do that comparison?
Right.
As a scientist, I want to have my, not placebo, but I want to have my base case. And is it different from the base case?
Right.
And it doesn't sound like they've got that here. So how's the agency making that determination, really?
It would be interesting to really dissect that trial design and maybe read more into it and understand what's going on, because, in light of this LDT rule, which seems overbearing on the industry, and then they're like, oh, this one's okay, we'll go ahead and approve it, it's fine. And the FDA is not perfect, and they're trying to get treatments to the market, and there's going to be different decisions in different situations, but you just kind of wonder how that's working. Now, if it works, it works, and I think I'm always excited to hear about these alternative types of treatments. So they're using electrical fields to disrupt cellular mitosis in cancer cells. So that's
cool.
that's kind of cool if it works, right?
Yeah.
Yeah, but one of my first reactions is always, whether it's chemical oncology treatment, it's what are you doing to normal cells, right?
Right.
So you're putting an electrical field there, and you always hear, don't live under high tension power lines, right?
Yeah.
So
Yeah.
We amplify that.
now you're saying
Now maybe we want to live under high tension
All right.
power lines.
but now you've got a cancer, and you're putting an electric field close to it. They say don't put your cell phone certain places on your body because of the electrical fields. So I've got a lot of questions, and I'm hoping that the agency is not just looking at this study, but considering what other external data there are around electrical fields, the relevance to how this particular device works, and how the treatments that it's being used with work, right?
Yeah.
And
Yeah.
can you see the actual additive benefit, before they say, yes, this is going to work.
Well, and you know that there's got to be loads and loads of in vitro data. So for our non-scientific listeners, right, work done in a test tube, right? So this would be so easy to test and retest and overtest, the actual work that goes on in a test tube, and see that it actually does work with the cells and whatnot. So
Could even be some animal models with.
Yeah. So I'm sure there's a ton more data out there that that's telling us something, and So, anyways, I think also if it's doing no harm, right, and if they're showing in the trial that there's limited to no side effects other than there's an additional procedure done, presumably to implant this thing, then any measure of efficacy would be something that I would want if I were a cancer patient. And oncology gets a special treatment in a lot of these situations because of the critical nature and the time-dependent nature. So I'm sure those are things that play into it that we just aren't, don't know, and it won't be written up in the news reports either. So great. Really interesting.
And it's a wearable device, so it's very non-invasive type of therapy. So yeah, if it, like you said, if it's doing no harm and it's showing some type of improvement, it's worth approving. It's worth studying and seeing what else we can do with it.
Yeah.
Excellent.
Very interesting. You come up with some very interesting science there.
Yeah, we loved it. We love doing the new news because it gets people thinking and it gets us thinking on our feet, and that's a lot of fun. So, Mark, when we were talking about what's been going on at AAPS this week, there were two themes that really popped out to me that I wanted to hit when I was here. One was the IVDR, because it's an area that I said IVDR. That's more the European versus the device, the IVD in the U.S. But anyways, but the other one was AI, and I did a talk that was about robotics and automation, and then there were, I think I counted over 20 talks just in bioanalytical about AI, and then even talks that didn't have anything to do with AI brought in AI. So what do you think about AI and bioanalysis and in the drug development in general?
Yeah. So I think AI is going to help revolutionize a lot of areas.
Yeah.
But I think people are looking at it as kind of a panacea. And I think that there are things that, if you heard the opening plenary session and they were talking about AI and some of the glitches it introduces, that you need experts to look at what it's done and really put it either in context or interpret what it is back into the real science. So I've talked to some of the companies that are looking to put AI into limb systems or reporting systems and doing quality controls and those types of things. Perfect places for it, right? Because these are rule-based systems, right? It's data checking, it's extracting information and transposing it. You know, it's great. It can do those types of things. It can do it faster than a human can do, and it can catch things that a human wouldn't catch. You know, there's 70,000 new viruses found in some research that was done recently and published, I think, last week.
Wow.
And they were all RNA type of viruses. And they were looking at them because they attack bacteria. And so here's a whole new...
That sounds good.
Right.
Yeah.
So
It depends on the bacteria.
Viruses attacking bacteria instead of developing other drugs that would be given to attack. So that could be a whole new area for what we're having with drug-resistant bacteria.
Right.
So, yes, could be a great new way for us to develop drugs. But somebody's going to have to take that, look at it, and figure out what is the potential for one of these to potentially also hit a human receptor, or an animal receptor, or a plant receptor, and all of a sudden it's attacking corn crops, You know, and so we can't just say it's the perfect solution.
Exactly.
We have to look beyond the particular impact area.
Because, you know, one of the areas I see AI as being really strong is in pattern recognition and finding patterns where we can't find them or it's very difficult. And so I can imagine with whether it's drug discovery, which I think tomorrow is the closing lecture is going to be about AI and drug discovery, which I'm really excited about, or identifying viruses that might attack certain bacteria, and we might not even know why it picks those, but it'll have some sort of a pattern recognition approach, or I have taken to taking spreadsheets of data, and I look at it and analyze it, and I just dump it into an AI, whichever one that you prefer, and it comes up with things and observations that I'm like, oh, I didn't see that, and so there's so many interesting different ways. Or Jim Shen was talking yesterday, just using it to program your robots, right? A really simple application. Hey, I want to do A, B, C, and D on my Hamilton pipetting robot, but maybe you're not an expert in programming. It'll give you the code. You might have to refine it, tweak this or that, dump it in. So there's so many different ways that are really exciting, overwhelming, actually.
Yes.
Yeah.
20 years ago, when the Hamiltons were coming out, our lab undertook an effort, and Muhammad Jamal was leading this.
Right.
We were trying to figure out, with solid phase extraction, how could we come up with a set of experiments for acids, neutrals, and basic drugs that we could screen a whole bunch of columns and come up with, this is the best approach to extract this drug out of plasma. And then go to the HPLC system, best chromatography as quickly as possible. Well, how do you program that into the Hamilton system? And get all of the permutations and all of those things. It was, an effort first to conceive all of it, and what are the criteria and those types of things, and what are the pHs and the solubility factors and all those things. Yes, an AI could probably have gone through in a day, given all of the appropriate literature, what that team spent 3 months trying to analyze, and then the team could just look back and, yes, we understand the chemistry and what they've come up with. Makes sense.
Yeah. So it's an exciting space to watch. It's an exciting space to watch, for sure. Yeah.
Indeed. What impresses me is just how fast the responses are when you give ChatGPT or whatever a glut of data and say, all right, make sense of this, summarize this for me, and ask it to give you some type of analysis, and it just starts right away, almost instantaneously looking at that and bringing it back. And so I think that's remarkable. And Mark, you mentioned earlier in this conversation on this topic that 70,000 new viruses were discovered and detected. Where are these new viruses coming from? Are these just mutations of other viruses? How does that happen?
These were just found in nature.
Just in nature?
Yeah. Okay. If I can find the article or where I saw it on LinkedIn, wherever, I'll get it to you.
Sure.
It's one of those things I saw, I read it, and I said, wow, we have another whole approach to dealing with drug-resistant bacteria.
Interesting.
That was really just stuck.
Fantastic. Yeah.
Excellent. So, Chad, any other questions from you?
I've had a great conversation. There were a few things I wanted to cover, but yeah, I think I'm good. Yeah.
Very good. So we have a tradition, Mark, that the last guest will write a question for the next guest, not knowing who that person is.
Okay.
So last recording, we had CEO of TrueLab, Scott Ogle, write this question, and then we're going to ask you to write a question as well for the next guest, not knowing who that'll be. We'll do that offline. So Scott asks, how do we encourage a risk-averse industry like ours to take a chance and innovate?
That is so hard when you're so regulated. I think successful groups have always been science-based, right? You can challenge the regulations if you're doing things on a solid scientific foundation, because the agencies will accept that, right? The questions when we first started using mass spec were, will the agencies accept it? Dry blood clots came out. Will the agencies accept it? And now there are 20 companies developing it. High-res mass spec, will the agencies accept it? All of these different technologies that are being used for PCR, for image-based cell analysis that are ongoing. These are all innovations that weren't in bioanalytics, say, that are now being applied because somebody said, I can use this technology. It fits the need, and the science is sound. Right? And just applying it. May not have the software that meets all of the compliance regulations, but the science.
So it sounds like, yeah. So if regardless of the technology, or maybe taking an existing technology and using it in a different way to generate some interesting results, as long as the scientific rigor. So encouraging those scientists, just keep rigorous with your science, use whatever technologies or invent new technologies that you think might work. And we've already shown a pathway to success through the agency, even though we're highly regulated. That's great.
And your biggest challenge is getting past your own regulatory departments who don't want to take it. Yeah.
Right. So they're going to say no.
Talk about what you want to do outside. Yep. Get your peers talking about it. And somebody else may get it to the agency first. But once you have that, everybody will follow behind.
And of course, as a service provider, probably the biggest challenge, one is funding, and two, getting it past the company that you're providing the service to, right? If you have a novel idea, you're trying to sell it to them, and then they have to sell it further on. So it's certainly a challenge, but I appreciate the pioneers and the innovators and the small companies that find funding, that drive things forward, that we keep this relentless pursuit of innovation and technology. And then you look back 20 years, we talked about antibodies earlier, the antibodies of 30 years ago, and we've moved so far since then. So innovation is happening. Feels like it could go faster, but we're risk averse for a reason as well, because we're talking about people's lives and curing disease. And so it's important to do no harm first, I guess. Yeah.
Right.
Indeed. That's great. Well, I will put a link in the show notes for everyone to reach out to Samaritan's Purse if you're interested in donating through them to hurricane relief that is very sorely needed. We'll also have these articles in the show notes and anything else that kind of comes through, and ways to reach out to Mark Arnold via LinkedIn and whatever other contact information he's willing to share at that point. Mark, thank you so much for joining us on site like this. This is really cool. I hope it was enjoyable for you, and it seems like a comfortable setting there. Thank you so much for being with us.
Well, thank you for inviting me. Great time. Always good talking to both of you.
Yeah,
We've known
likewise.
each other for a long time.
Yes, we have.
Yeah.
Yes, we absolutely have. And likewise, thank you, Mark. And of course, we said it before, but thanks to APS for hosting us,
Indeed.
letting us do this with our passion project of BioTalk on Zip. This has been great.
Thanks a lot.
Thank you.